Assalamu alaikum students. Welcome to the course of plant biotechnology. Well, this might be our very first class where we can be introduced to each other in person. But due to the pandemic of COVID-19, we are starting the semester in a different way. We have to go online in order to take precautionary steps. However, this recording is for the students who will not be able to join my Zoom class. So this can be used as a backup for all of you. So I'm not taking too much time in introducing myself, as I think by this time all of you somehow familiar with me by any of your previous courses. So let's start talking about the course. BTE 308. This is one of your core course. Before that, you have learned about plant and animal in your BTE 103. From there, you have a basic idea about plant biochemistry or some other necessary reactions regarding plant morphology and regulation. Here in this lecture, besides learning about some plant morphology and some other regulatory aspects about plant science, we will focus on the biotechnological part of plant science. That means how we can apply plant or agricultural science in case of biotechnology. In my first lecture, I will give you a very brief history about how this term or this aspect of biological science has evolved. What are the basics of plant biotech? And of course, I will let you know some important discoveries that will help us to establish biotechnology using plant cells or whole plants or whatever it is, any other plants for our own purpose. So let's look at the screen. This is our title of the first lecture, which is Plant Agriculture, the Impact of Biotechnology. History. So what we know about plant biotech, how it is evolved, from which period we started to learn about that plant cells can also be used in case of technological purpose. We can engineer a plant cell or we can modify a plant cell in order to get some commercial product. The basic principle regarding this is cellular totipotency and genetic transformation. These two words or these two phrases, cellular totipotency and genetic transformation, is the main foundation to establish plant biotech. And these two things were tracked by two scientists. One is Theodore Swan and another is Griffith. And Theodore Swan was not alone. Another scientist, Sladen, also helped him in order to understand or know about cellular totipotency. In the next slide, we'll know what is cellular totipotency. And about Frederick Griffith, I think by this time you know that Griffith conducted an experiment using two strains of Streptococcus bacteria in order to show that bacteria or microorganism or living things has the capability of uptaking a foreign DNA, and not only uptaking but also to acquire the traits regarding that foreign DNA. So in the 18th January 1983, that means almost more than 25 years ago, it is arguably the most important dates in the history of plant biotech. Let's see why. Because in that period, at Miami Winter Symposium, 3 independent groups described Agrobacterium tumefaciens mediated genetic transformation. That means 3 independent groups worked with Agrobacterium tumefaciens, which is a plant bacteria causing a disease or a crown gall disease in plant root, that these bacteria or this mechanism of bacteria causing root disease can be used as a DNA delivery or genetic transformation method, and that was a revolutionary discovery. After that, the whole scenario of plant biotech has been changed in a drastic way. So what they did, they actually find out that Agrobacterium tumefaciens mediated genetic transformation can lead to the production of normal fertile transgenic plant. That means through this process, through this approach, they can produce a transgenic plant which is a normal and of course fertile. That means this transgenic plant can be used easily in order to generate or in order to proceed towards many more population. It is totally normal and fertile. Now let's see what is cellular totipotency, which is one of the bases of modern plant biotech. Totipotency or cellular totipotency is the ability of a single cell to divide and grow by itself in order to form a whole plant. That means when we put a single cell in a proper nutrient condition, and if that cell will divide by itself, ultimately having the whole structure of a whole plant, like having stem, root, leaves, and all other organs, then we can say that yes, that cell has the totipotency capability, or that cell is totipotent. And about genetic transformation, all of you know that it is either genetic modification of a persistent DNA or uptake of a foreign DNA in order to have a novel characteristics or novel valuable characteristics. So these two are the most important terms that we must learn in order to understand the plant biotechnology. So totipotency of the plant cell were experimentally proved by a Hungarian and German botanist. Basically, that botanists were born in Hungary. He was an Hungarian by born, but he was brought up in German. And his name is Haberland. You can see here. His name is Haberland. Was the first to try to obtain experimental evidence of totipotency by culturing plant cells. Look, he cultured plant cells in a nutrient solution in the hope of regenerating whole plants. However, his experiment was totally failed, and let me tell you why and what are the blunder of his experiment that never let him to succeed. So he presented the results of his pioneering experiment in a prophetic and now classic paper entitled Experiments on Culture of Isolated Plant Cells. So he did the experiment in proper way. He also published a paper regarding this. The title here is Experiments on the Culture of Isolated Plant Cells. However, he did some major blunder, or some major obstacles were arise when conducting his experiment. What he took different types of cells from different organ of a plant. From leaves, he took differentiated cells containing chloroplast. He also took petiole cells, that means petals, cells from petals from another species of plants, glandular here, and also stamen here. That means he tried to have a pure and sophisticated experiment by having different types of cells. And the solution he used was called Knopf salt. It was one of the renowned plant solution or nutrient solution at that time. However, he failed. Now let's see why he failed. He failed largely because of his unfortunate choice of experimental material. Although he took different types of cells from different organelles, organ of a plant, but he never took the exact one. At present, we all know that it is the cell of a tip, either apical meristem or apical root tip. From a tip, we have to take cells in order to let it grow, or in order to generate callus or a whole plant from it. Otherwise, cellular totipotency or dividing capacity of other cells were not that much as a root tip or tip cells, apical meristem, whatever it is, the tip cells. We have to take cells from there. He never took cells from there. That it was one of the major reason of his experimental failure. And another reason was the solution or nutrient provider he took was not adequate. It was not enough to provide enough nutrient media in order to let that single cell to grow in a whole plant. So these are the reason of his experiment to fail. However, he tried, or his hypothesis is a pioneering work. He first tried to show that yes, plant cells can grow into a new cell. However, he took a wrong type of cell. He cannot choose the better cells, but his theme and his experimental approach were correct. The material he chose was wrong. Now, the development of improved nutrient solutions. Well, as I said, that Knopf solution was not adequate in order to let a single cell to grow in a whole plant. Then properly developed nutrient media were discovered or identified. So what should be the constituents of a improved nutrient solution? Plant growth regulator indole 3-acetic acid. We know that it is a very important growth regulator for any plant to grow. Informed choice of plant material. That means from which type of cell we have to choose as our culture seed. Appreciation of the importance of aseptic cultures. That means while culturing, like while inoculating the plant cell, taking a petri dish, letting it grow, all things or all culture environment should be aseptic, so that no other extra contamination can occur here or can contaminate the plant culture. led to sustained growth of root tips and carrot root and tobacco stem tissues were identified for indefinite periods of time. That means led to sustained growth of root tips. While they have used or the using of such media led to the sustained, that means persistent growth of root tips and carrot root and tobacco stem tissues for indefinite periods of time. That means from a longer period of time. This important milestone was achieved by Roger Guthrie and Pierre Renaud in France and in United States. That means both in France and United States, such experiments were conducted using plant cells with the hope that scientists can grow a huge plant or a whole transgenic plant from a single cell. Then comes the discovery of plant growth hormone. We will discuss about it in our next portion.